We move further in the topic. Now the functions of helper T cell. We move on with the topic, the functions of helper T cells. Helper T cells share the following common features with cytotoxic T cells. Their T cell receptors are made up of alpha and beta chains. Cytoplasmic tails of T cell receptors is associated with similar CD3 complex having items. They have co-receptors associated with LCK. Intercellular signaling occurs by a cascade of phosphorylation reactions involving tyrosine residues. The helper T cell differ from cytotoxic T cell in these following respects. The co-receptor is CD4 made up of a single polypeptide chain folded into four extracellular domains, D1 to D4. The joint between D2 and D3 domain is flexible. The T cell receptor recognizes a foreign peptide bound to MCH class 2 proteins. Now we look upon antigen presenting cells. The MCH2 proteins are present on macrophages, dendritic cells, and B lymphocytes. These cells are known as antigen presenting cells or APCs. The T cell receptor of a helper T cell binds to the specific antigen fragment displayed by MHC class 2 proteins of antigen presenting cells. The domain D1 domain of CD4 binds to beta 1 domain of MHC2 protein. LCK becomes active and phosphorylates the items of CD3 complex. The ZAP70 binds to phosphorylated items and phosphorylates the residues of tyrosine of some target proteins. This results in the release of cytokines or lymphokines from helper T cells. Diagrammatically, we can see here the antigen presenting cell along with MCH class 2 protein and all its unit alpha 1, alpha 2, beta 1. We can see the CD4 and the helper T cell. We can see the antigen fragment trapped between alpha and beta of MHC2 protein and T cell receptor. We can see the adjoining CD3 complex along with CD4 complex. We can see the further chains of proteins down here and items including LCK. And now the topic moves to lymphokines. Lymphokines includes interleukins, which are from interleukin 2, 3, 4, 5, 6, etc. Granulocytes, colony stimulating factor or GCSF. Granulocyte macrophage colony stimulating factor or CSF. Tumor necrosis factor or TNF, alpha interferons or IFN. Actions of lymphokines. The IL-2 stimulates multiplication of helper T cells so that more helper T cells are available to mount an immune response. Interleukin 3 stimulates the proliferation of bone marrow stem cells. Interleukin 4 stimulates the proliferation of B cells, T cells, and mast cells. Interleukin 5 stimulates the growth of eosinophils. Interleukin 10 stimulates the growth of mast cells and increase the synthesis of MHC class 2 proteins in B lymphocytes. Interferon activates macrophages and natural killer cells or NK cells. TNF alpha activates macrophages. GM-CSF stimulates the growth of granulocytes and macrophages and also stimulates the differentiation of B cells and dendritic cells. Some lymphokines attract the phagocytic cells to site of infection. Thus, release of lymphokine from helper T cell leads to increase in the number of helper T cells stem cells, increase in the number of cytotoxic T cells, increase in the numbers of B cells and plasma cells, increase in the number of antigen presenting cells, increase in the number of phagocytic cells, activation of phagocytic cells. In this way, both innate and adaptive immunity are geared up to fight against the pathogens. The overall result of these actions is that an effective immune response is mounted to meet the challenge posed by the antigen or pathogen. Helper T cell responds to call the help given antigen presenting cell in the form of an antigen fragment displayed on their surface. Helper T cell do not destroy the antigen themselves. They respond by activating all arms of defense system and coordinating their actions so that threat posed by antigen is effectively met. Helper T cells are therefore described as the commanding officer of the defense forces. If helper T cells are depleted or debilitated, the entire immune system is crippled. Functions of suppressor or regulatory cells. They play a regulatory role in immune response. They suppress the activity of helper T cells and cytotoxic T cells. They prevent a disproportionate immune response. They help to prevent immune response against self molecules. Functions of memory T cells. Memory T cells retain memory of the antigen after the first encounter. They spread throughout the lymphoid tissues. They are released quickly into circulation if the same antigen enters again. They also help in mounting a quick immune response. Self-tolerance. Many molecules have antigenic properties and properties are present in our body. Also, our immune response does not act against self molecules, which is known as self-tolerance. Self-tolerance is partially due to the action of suppressor T cells, but there is a second and more important mechanism. The newly formed lymphocytes are processed in fetal life before they reach the lymphoid tissue. The purpose of this processing is twofold. The first one is to allow only those lymphocytes to mature that are immunologically competent. And the second one is to eliminate those lymphocytes that can react with self antigen. The processing of T cells include developing T cell express T cell receptors or TCR, which are known as CD4 and CD8. Some of the TCRs are incapable of recognizing any self-MHC protein, and such T cells would be functionally useless if allowed to mature. Therefore, the developing T cells are first subjected to positive selection. If the TCR and CD4 bind to some MHC1 protein, the cell is allowed to mature into cytotoxic T cell and expression of CD4 is suppressed. If the TCR and CD4 bind to some MHC2 protein, the cell is allowed to mature into a helper T cell and expression of CD8 is suppressed. The positively selected cells are then subjected to negative selection. The T cell that binds self antigen, self MHC complex is undergo apoptosis, which is known as the programmed cell death, and thus they are eliminated from the system. A processing of B cells. Developing B cells that are capable of recognizing self antigens can have four different fates. First is receptor editing, clonal deletion, energy, and immunological ignorance. Developing B cell that encounters multivalent self antigens undergo further light chain gene rearrangement until the antigen receptor is modified that cannot bind the self antigen. In clonal deletion, if self reactivity is not lost by receptor editing, the developing B cell undergoes apoptosis. Thus, the entire clone of the self reacting B cell is deleted. In energy, developing B cells that encounter self antigens of low valency becomes energic. Their antigen receptor remains within the cell and signal transduction is impaired. Immunological ignorance. If the self antigen is segregated in a compartment where B cell cannot reach, or if the self antigen is present in insignificantly low concentration, the B cell capable of reacting with them will mature but will remain immunologically ignorant. Thus, developing T cells and B cells are processed in such a way in fetal life that the cell capable of mounting an immune response against self antigen are deleted or modified or incapacitated. This results in self tolerance. However, self tolerance is sometimes lost, resulting in autoimmunity. Now we see autoimmunity or autoimmune diseases. If the immune system reacts against a self antigen, it results in an autoimmune disease. Autoimmune disease can occur because some self molecule combine with microbial protein to form new antigen, which are treated as foreign antigen by the immune system, or some self molecule having a close structural resemblance with foreign antigen may be targeted by the immune system. Some diseases resulting from autoimmunity include Hashimoto's thyroiditis, myasthenia gravis, systematic lupus erythematosus, rheumatoid arthritis, some cases of type 1 diabetes mellitus, male infertility, and pernicious anemia. Immunodeficiency, it occurs due to impairment of the immune system. It may be inherited primarily or it can be acquired, which is secondary. Inherited immunodeficiency may affect humoral immunity or cell-mediated immunity or both. Congenital A-gamma globulinemia is an X-linked disease in which only humoral immunity is impaired. DiGeorge syndrome occurs due to lack of development of thymus because of which cell-mediated immunity is impaired. Several combined immunodeficiency diseases or SCIDs occur from deficiency of adenosine deaminase because of which both humoral and cell-mediated immunity are impaired. Acquired immunodeficiencies may result from severe malnutrition, irradiation, immunosuppressive drugs, or viral infections. The most common among them is infections with human immunodeficiency virus or HIV, which causes a severe disease called acquired immunodeficiency syndrome or AIDS. HIV is a retrovirus having an RNA genome. Its surface protein GP120 is complementary in structure to CD4 protein of helper T cell. Therefore, HIV binds avidly to helper T cells. We can see the structure. Here, the helper T cells along with its GP120 protein and T receptors. What happens that when genomic RNA along with reverse transcriptase and protein and HIV, this is the HIV virus, it has all these things, genomic RNA, reverse transcriptase, the GP120 and HIV, and when it infects a cell, it forms its own nuclear material which replicates to form a DNA, which further replicates to form different proteins, and thus obviously which is again a second virus, and this virus again uh, proportionate it divides itself and produces its copies. Multiplication of HIV inside a helper T cell leads to rupture of the cell. Depletion of helper T cell cripples the immune system. Allergy is described as a side effect of immunity. It is an alternate immune response to an otherwise innocuous antigen. Antigen evoking an allergic response are called as allergens, and allergens are small molecule present in pollen, dust mite, drugs, foods, pets, and etc. Allergens bind to the immunoglobulin E, reagenic antibody. IgE is normally present in minute concentration and protects against parasite. Some persons have a relatively high level of IgE, influenced by genetic as well as environmental factor. IgE binds to high affinity receptor of the mast cells. Mast cells are present beneath mucosal cells and in connective tissues. When an allergen binds to IgE, the mast cell release their stored granules containing some chemical mediators. The chemical mediators include histamine, heparin, leukotrienes, CD4, D4, NE4, platelets, activating factors, eosinophil, chemotactic factors, etc. These chemical mediator causes increased in capillary permeability, vasodilation, itching, sneezing, bronchospasm, etc., depending upon site where the allergic reaction has occurred. The allergic diseases include allergic dermatitis, allergic rhinitis, urticaria, bronchial asthma, drug allergies, food allergies, etc. With this, we conclude our topic of immunochemistry, last being allergy, which is a side effect of immunity basically. And it occurs in persons having a high quantity or high measure of IgE.